In this lesson, we're going to look at the chemistry of life. And to start with, we're going to look at organic molecules. Remember that organic refers to molecules that have both atoms of carbon and hydrogen. The organic molecules include carbohydrates, lipids, proteins, and nucleic acids. If we look at carbohydrates, their function is to provide quick energy. It's easy for the body to use, and we need it in its simplest form. The building block is a monosaccharide. This hexagon shape is seen here, and that we refer to as a simple sugar or just sugar sometimes. The chains that are formed are called disaccharides and polysaccharides. Saccharide being sugar, mono means one here, di two, and poly more than two. And monosaccharides are going to be in a 1 to 2 to 1 ratio of carbon, hydrogen, and oxygen. If we look at this molecule here, we can see what a disaccharide looks like. We have two of these simple monosaccharides joined together. If it were a polysaccharide, then we would have more as well. If we see the term "-ose" as a suffix on a word, it refers to sugar. So if we look at monosaccharides, okay, which are simple or single sugars, their chemical formula is C6H12O6 in that 1 to 2 to 1 ratio. And we see the terms glucose, fructose, and galactose. Disaccharides are two sugars, two of those monosaccharides joined together, and we'll see a formula like C12H22O11. Examples include sucrose, which is actually a molecule of glucose plus a molecule of fructose, lactose, which is glucose plus galactose, maltose, which is glucose plus glucose, and polysaccharides, which means many sugars, very complex. We look at starch, which is found in plants as a result of photosynthesis, and it's how plants store that excess sugar. Cellulose, which is also found in plants, and that helps them form cell walls. And glycogen is a complex form that we find in humans that's stored in muscle and liver cells. The next molecule is proteins. And the building block of proteins is amino acids, which is also called a peptide. So if we join many amino acids or peptides together, we form a polypeptide or protein. The number and sequence of these amino acids determines the protein's shape and job. Looking at the structure of an amino acid, we can see that there's a central carbon, a functional group, hydrogen group, or carboxyl group, and an amino group. Note that in the amino group you see nitrogen. This is unique to proteins. Proteins have many functions. They help build structures such as muscle, hair, nails, skin. Some of them function as enzymes and control the rate of chemical reactions. Some of them are in the blood as antibodies that fight bacteria. Some are in the blood as hormones that are chemical messengers. They serve as a backup energy source when carbohydrates and fats are depleted. This is a very backup energy source. People that unfortunately are undergoing starvation may be breaking down their proteins for energy. And some of them are located on the surface of the cell membrane to receive messages from the environment, and these are called receptors. Proteins are also very sensitive to environmental changes, such as changes in pH and temperature. And if the shape changes, becomes unraveled, the protein can't work. If a protein is exposed to the extreme pH and temperature, it is said to be denatured or has changed in shape. The next molecule is lipids. They are nonpolar, which means they do not dissolve in water. We say they are hydrophobic, hydro, water, phobic, fear of, because they do things like sit on top of the water if you think about salad dressing, how that separates, or the lab that we did with the oil and the water. They do not combine. There's three types of lipids: fats, phospholipids, and steroids. If we look at fats, okay, this is a structure of fats. It's a type of lipid that we eat. It serves as a backup source of energy to carbohydrates. It maintains insulation to help hold stable temperatures, and it protects and cushions organs. They're called triglycerides because the building block is glycerol and fatty acids. Triglyceride because there are three fatty acid chains. If we look at this molecule down here, we see that this is the glycerol backbone, and then each of these is a fatty acid chain. We also call lipids hydrocarbons because they are composed of large amounts of just carbon and hydrogen. There's three types of fats: saturated, which are solid at room temperature and typically are animal-based, such as butter, meat, and cheese. Unsaturated fats, which are liquid at room temperature, and these are plant-based, so we find, unsaturated fats in olive oil and vegetable oil. And trans fats, which are unsaturated fats that were hydrogenized, which means that we added hydrogen into like margarine and Crisco. They're very unhealthy and they increase the risk of obesity and heart disease. Phospholipids form cell membranes, and they're known as diglycerides because they're made up of a glycerol and two fatty acid chains. If we look at the structure of a phospholipid, this would be a representation of a phospholipid bilayer, having two layers. You have a phospholipid head and two phospholipid tails. The phospholipid head is actually hydrophilic, meaning water-loving, and the tails are hydrophobic. Looking at steroids, there's two types. The first is cholesterol, which can come from some foods. It gives cell membranes their flexibility, but it can clog arteries. The other type is steroid hormones, which include estrogen and testosterone, the female and male sex hormones. Nucleic acids are the last organic molecule we're going to look at. They can carry genetic information and the code to make proteins. There's two types: DNA, which is called deoxyribonucleic acid, and RNA, which stands for ribonucleic acid. The building block is called a nucleotide, which includes a phosphate, a 5-carbon sugar, and a nitrogenous base. There are 4 possible bases in each DNA and RNA. If we look at DNA, we find it in the nucleus of the cell, and it controls the production of proteins within the cell. The structure is a double helix, like a twisted staircase, and that was found by James Watson and Francis Crick. The backbone of the helix consists of phosphate and deoxyribose, the 5-carbon sugar. So if we look at this model here, we can see our 5-carbon sugar and then our phosphate group. And the bases connect according to the law of base pairing. So adenine is going to match with thymine, and cytosine is going to match with guanine. Okay, so they're going to pair specifically here. The strands are said to be antiparallel, which means one backbone is upside down so that these shapes fit. And the order of the bases determines the instructions for the proteins to be made. If we look at RNA, it's a single strand. It is the blueprint of that master copy DNA that takes the message to the ribosomes outside of the nucleus. So this copy of the DNA would be made in the nucleus and then it travels to the ribosomes in the cytoplasm. It is made up of 5-carbon sugar called ribose, and it has 4 bases as well: adenine and uracil, cytosine and guanine, and these match up just like they do in DNA. Enzymes. Enzymes are a type of protein that speed up chemical reactions. They're called catalysts. Their name ends in ase. So sucrase is the enzyme that digests sucrose. Makes it easy to identify many enzymes. And enzymes are going to attach to a substrate. So if this is our enzyme down here, then this one is going to match specifically to a substrate, and that's based on shape. It is very specific, and sometimes we call it a lock and key, but we also use the induced fit model. So when the substrate and the enzyme combine, the lock is really going to be made and it will seal. The enzyme and the substrate match up, and the substrate fits into the enzyme at the active site. When this happens, then the reaction can take place. After the substrate detaches, then the enzyme can be recycled. And remember that enzymes can either synthesize, build, or digest, break down molecules. After they work on the one molecule, then they are reused and they work on another. So if we look at this reaction, we have the enzyme with its active site, and it combines the substrate. We have the enzyme substrate complex, and then the enzyme breaks down the substrate into 2 products. This can go the reverse and take the 2 products and make it into one. If we look at what can affect enzyme rate. If we decrease the amount of substrate, the reaction is actually going to slow down. If we increase the amount of substrate, it's going to speed up. Same thing if we increase the amount of enzyme. If we decrease the amount of enzyme, the reaction will slow down. And if we change the pH, the reaction will also slow down. And temperature is very interesting because as we increase temperature, the rate of the reaction will actually increase until the temperature gets to a point that's too high and the enzyme denatures. Because it is a protein, it unravels and can no longer work. So if we look at how these monomers become macromolecules or polymers, we have a reaction called dehydration synthesis. So what happens is we take two hydrogen atoms and one oxygen atom, otherwise known as water, so that a bond can form between the monomers. If we look at this monosaccharide here, and here we have two glucose molecules, we take away two hydrogen and an oxygen, and then a bond can form here, which is maltose. And that forms our bond. That is, so dehydration synthesis means taking out a water molecule so that the bond can be formed. If we digest it, it's a reaction called hydrolysis, where a molecule of water is added to the bond to break it apart. If we take this sucrose molecule and we add water to it, now we can break it down into its monomers, glucose and fructose. In comparison, hydrolysis is breaking down or digesting molecules from polymers, and it adds a water molecule. So we add water to the side. Dehydration synthesis builds larger molecules. They go from monomers to polymers by removing a molecule of water.